Anybody ever been to the Washington Cathedral? And this is the north rose window in the Washington Cathedral. The reason I'm showing this is even in Washington, D.C., if you look hard enough, you can find something beautiful. If you look hard enough. Oh, there it is. That's not a stained glass window. That is a computer-generated picture of a cross-section of DNA. So what we're looking at in this picture is the view of DNA from this angle. We're looking down the barrel of the DNA. So imagine you're at the top of a spiral staircase looking down. Okay? That's the view. And that's a computer-generated picture there. And each colored dot that you see is an atom, or represents an atom, each colored dot. So there's beauty in nature if you look hard enough. How do we know DNA looks like this and is built like this? How do we know? Well, back in 1953, and those two guys right there are they've got most of the credit for the discovery. They determined this three-dimensional structure of DNA working at Cambridge University in England. They discovered this three-dimensional structure of DNA back in 1953. The guy on the left is James Watson. Oh, trivia question. Does anybody know where James Watson got his PhD? Purdue. No, not Purdue. Sorry. Not MIT, not, okay, I'll tell you the answer. Indiana University. Which just goes to show you that nobody's perfect. So Watson and Crick, and they worked with this other scientist, Maurice Wilkins. I always tell my students, scientists hardly ever work alone. It's a collaborative effort, and you better learn how to work in groups if you want to be a scientist. But anyway, Maurice Wilkins. Now, those three, Watson, Crick, and Wilkins, because of their work, they received the Nobel Prize in Physiology or Medicine in 1962 for discovering the three-dimensional structure of DNA. That it looks like that, it's built like a twisted ladder, and we'll look at the parts in a little bit. Now, there was another person, Rosalind Franklin, brilliant scientist. She was at King's College in London, and a brilliant X-ray crystallographer. Probably one of the absolute most gifted scientists in the bunch. But she had a piece of the puzzle that she had discovered. You see, Watson and Crick were really struggling, trying to figure out the three-dimensional structure of DNA. They were getting close. They were missing vital pieces of the puzzle. And she discovered the vital missing piece of the puzzle, and it was like lying on her desk, okay? Now this piece of the puzzle is famously called Photo 51. That's the vital missing piece of the puzzle. It was unpublished, and Watson and Crick used it without her permission. So, you know, there's been that debate, did they steal it? Well, they used it without her permission. Now, when the three, Watson and Crick and Wilkins, received the Nobel Prize in 1962, Rosalind Franklin was not up on the stage with them. She did not share in the Nobel Prize because, sadly, she died in 1958 at the age of 38 from ovarian cancer. And you can't win a Nobel Prize after you die. So she's getting her proper due nowadays, and I emphasize her in my classes with the kids. And their heart goes out to her, as it should, and she should be given so much credit. It was her missing piece of the puzzle that helped them solve the problem. This model right here, 16 base pairs long. Okay? And I got to thinking one day, if we wanted to make your DNA the size of this model, okay? All the DNA packed inside your 46 chromosomes in just one of your cells. If we wanted to make your DNA model, your DNA this size, how much longer would this have to be? Really long. Okay, I calculated and I used ChatGPT to help me, correct me if I was wrong. You see, in all the DNA in your 46 chromosomes, you've got 3 billion base pairs. And so to incorporate 3 billion base pairs, this model, this size, if your DNA was this big, would stretch around the world almost 3 times. Okay? That's what? Macro. No wonder we call it a macromolecule. It just blew me away when I calculated it. And I thought, can that be right? So like I said, I had ChatGPT do the calculations too. I can't always trust it 100% of the time, but it worked pretty well. You see those lines? Those are like forces of attraction. It's almost like T and A like each other, and C and G like each other. They're attracted to each other. Now those lines, those forces of attraction between them, they represent something that are called hydrogen bonds. And they're very, very weak forces of attraction, which is a good thing, because we're going to see here in a little bit that DNA has the ability to split apart the two sides in order to kick off the process of what we call DNA replication. And so those hydrogen bonds holding those bases together, they're very weak, and that's a good thing. Here's what we're talking about. Here we have a T and an A, those two bases joined together, and down here a G and a C. Now you see that oxygen atom right there? I don't know, can you all see that? The green dot? Look at that, that's what happens when you have 2 cups of coffee. That oxygen atom right there sticking out on this thymine. Yeah, it's sharing electrons in the rest of the thymine molecule. It's sharing electrons, but it's an unequal sharing. It's kind of like you have a husband and a wife in bed, and it's a cold night, and in the middle of the night the husband is shivering because the wife is curled up and has most of the blanket around her. You know what I'm talking about? And so they're sharing the blanket, but it's kind of an unequal sharing. That's what's going on here. That oxygen atom, yeah, it's sharing electrons with its neighbor on this thymine molecule. This oxygen atom is sticking out. But since it hogs most of the electrons that are around it, it has a slightly negative charge. On the other hand, over here on this hydrogen atom sticking out on this adenine molecule, it's sharing electrons with this nitrogen atom, of course, but nitrogen's hogging most of them, most of those negatively charged electrons, leaving that poor hydrogen atom that's sticking out on the end there with a slightly positive charge. And so what happens is that oxygen there has a very slight negative charge, and that nitrogen there has a very slight positive charge, and opposites attract, and so they're kind of pulled together just a little bit. It's a very weak force of attraction. That's called a hydrogen bond. And you can see there, between T and A, there's 2 hydrogen bonds, and between G and C, there's 3 hydrogen bonds. I used to tell my students during passing period in the hall, when I'm standing out there and I'm watching students in the hallway, high school kids, going to and from classes. They have 5 minutes in between classes. There's a lot going on, a lot of social dynamics. And I tell my students, sometimes I'm standing there watching and I'll see somebody acting like an A and another person acting like a T, and it's obvious they're attracted to each other. I can tell just by watching. I've watched mating behavior in the halls for 42 years. I can recognize. And sometimes over here there'll be two kids playing the roles of the C and the G, and they're attracted to each other. It kind of works that way. Now let's look at a really neat process. It's called DNA replication. DNA really does two main things. It takes care of you for your entire life. It controls all those genes. It controls the manufacture of all the protein molecules that make up your body and run your body. And next time we're going to look at that, how that works. But the other thing DNA does that's real important is it replicates. It can make copies of itself. And that's what I want to look at. Now, to illustrate this, see, I've got these magnetic models that I made, and I didn't want to assume that I would have a magnetic or a chalkboard to stick them on. So we're going to go ahead and watch this. I prepared this. It's an 8-minute video clip that illustrates DNA replication that we can watch together. Okay? And I think you'll see how DNA makes copies of itself. The neat thing about DNA is it's able to make copies of itself. DNA is able to replicate itself. That process, of course, as you might imagine, is called DNA replication. Now, sometimes when DNA is replicating, it makes a mistake, or the enzymes responsible for replicating the DNA might make a mistake. Okay. And if that happens, that mistake's called a what? Mutation. A mutation. Okay, very good. It's amazing. 100 million base pairs without a single mistake. Here's what we're talking about with exons and introns. I'm going to read that gene. I'm going to read it out loud. Y'all ready for this? Now, on the front end of this gene, this sequence of DNA, there's a promoter. That's a little like on-off switch. Genes are more involved than we're getting into. That's an on and off switch. So let me read the instructions in the DNA from left to right. Here we go. Oh, remember, the blue pieces are introns. They're part of the message in the DNA that's going to be cut out and thrown away. All right, here we go. Let's read it. Okay, ready, begin. That was the promoter region of that gene speaking. Okay, here we go. May. So far the message is May. May? What? Let's read this first intron. That's just garbled A's and T's and C's and G's that don't mean anything. We're going to edit that out, don't worry. Let's move on. All right, so far we've got make. Blue eyes. What was the message? Make blue eyes. Now, it's kind of like that. It's kind of like that, okay? All right. This is how our genes actually work. Now, just out of curiosity, I looked up the DNA sequence online of the gene that when it mutates causes cystic fibrosis. And here's the DNA sequence of the gene, the normal gene. And I know you can't read this, you're not supposed to. A, T, G, and so on. All those letters make up the DNA sequence of the gene that, when it goes wrong, causes cystic fibrosis. And remember, the only difference between this gene and the bad one is 3 letters are left out. Causes cystic fibrosis. Now that's not all of it. There's the rest of it on the second page. Now, On those 2 pages, we're just looking at the results of the exons. There were 98 pages of introns that I did not print out. So that gene is almost all introns. That's kind of how it's put together. An E. coli bacterium will replicate all its DNA, divide in 2 to make 2 bacterial cells, and then 4, and then 8, and then 16, and 32, and 64, and 128, and 256, and so on. That divide every 20 minutes. So you could theoretically start with 1 E. coli bacterium in a test tube of beef broth in the morning. In the late afternoon, you've got billions of copies of that one bacterium, and they're all genetically identical. So very useful tools. So we have the tools. Back in the early 1970s, this is pretty much worked out in the early 1970s, biologists sort of said, we've got the tools that we need now if we would like to take maybe a human gene, put it into a bacterial cell, and get that bacterium to read that human gene and produce whatever protein that human gene specifies. If you put, let's say, a gene that controls the manufacture of a very important protein called insulin, and you splice that gene into a plasmid in the laboratory, and you can convince a bacterial cell to absorb that plasmid, that bacterium is going to read that little human insulin gene, and if it says make human insulin, that's what it's going to do. It's all the same language. Bacteria know how to read A's and T's and C's and G's. DNA is DNA, it's all the same stuff, and it's going to make human insulin. You see where we're going with this? Now, those are the tools. So biologists got real excited in the early 1970s. They said, you know, we've got the tools now. We can do this.
We had the tools, we had the talent.
Got the tools and the talent. Okay? Now, let's look at how it works. Recombinant DNA technology. You've got this picture on the top of page 2. Let's look ahead here. Okay, now I've got 3 real people. Mo, Larry, and Curly. Okay? Of course, they're not around anymore, but when I was a kid, we would watch them. And my mother was so worried that my siblings and I would imitate them. And we never did. We kind of did better. But let's compare their 3 DNA samples here. So let's look at the parts. Okay, there's a gel. A gel is like a slab of almost clear jello. A slab of almost hard, clear jello. Okay? And the wells you saw in the video, how they use the comb when they pour, it's like making jello. When they poured the liquid gel into the casting tray, they had a comb in there that when they pulled it out after the gel was hard, it left these holes or these wells. Okay, that's the gel. And then if you look here, those are the wells. Okay? And those are the parts. Now, let's take Mo's DNA first. I assume everybody knows who Mo is, right? Let's take Mo first. Okay. I've extracted the DNA from Mo, and I've added a restriction enzyme to it. And let's pretend that this string of red yarn that goes from here to here, that is Mo's DNA. It's part of non-coding DNA, and we're going to compare the same stretch of non-coding DNA in Mo, Larry, and Curly. Okay? We add the restriction enzyme to Mo's DNA, and okay, here's the restriction enzyme. The restriction enzyme is coming along here, and it's looking for a GAATTC. And it comes along, let's see, is there any GAATTC? No. Oh, there isn't any. So we're going to end up with how many fragments from Mo? Just one. All right, let's see. Maybe we can add his DNA then to the well. There it is. Okay, now let's look at Larry's. Okay, here's Larry's DNA. And the restriction enzyme is coming along here looking for a GAATTC. And, whoa, there's one right there. There's a cut site. So we get how many fragments from Larry? 2. A long one and a shorter one. Okay, so we'll add Larry's DNA to the well. Okay, let's look at Curly. Now Curly is the one that probably cuts up the most. So now we're going to look at Curly's DNA. Curly, what do you know? He's got the same restriction site here that Larry has, but lo and behold, there's another one right there. So Curly's got 2 cut sites. There's a GAATTC here, and there's a GAATTC here. So we get 3 fragments, right? So we're going to load Curly's DNA into his well. Now, we're going to hook this up to a power supply. There's buffer solution. It looks like water. It looks like water, but it's got ions and salts in it. And so that gel is totally submerged in this gel box. It's got buffer solution and it's totally submerged. And what that buffer does, the ions in it help for the electric current to move through that solution. And also the salts in there prevent the pH from jumping all around while the current's turned on. Okay? So we position those wells so that they're nearest the negative electrode. The DNA is negatively charged. The DNA molecules are negatively charged. So you turn the current on, and they're going to start migrating through the gel towards the positive electrode. Like this. Like that. And the bigger fragments have a harder time migrating through the gel, so they're slowed down. They don't go very far. The shorter fragments, they slip through easier, so they move farther and faster through the gel. And if you remember, Mo had 1 fragment, Larry had 2, Curly had 3. That's kind of how it works. The reason, now DNA is a molecule, right? You're thinking, how do you see it? But the reason you can see a band here is because we've extracted millions of cells from Mo, and they're all genetically identical, millions of cells from him. So we've got millions of copies of that fragment, and they all accumulate here because they're all the same size, and that's how come that band is even visible. All right, DNA fingerprinting. Let's look at some practical uses. One of the practical uses is called pre-symptomatic diagnosis. Now, time out. This method of DNA fingerprinting we're looking at using what are called RIFLIPs, restriction fragment length polymorphisms, is the earliest method developed of DNA fingerprinting. There's a fancier, in some ways easier, method of doing DNA fingerprinting that we're going to get into on Wednesday. But I wanted to get this because it's kind of the basics. Now let's look at pre-symptomatic diagnosis. Okay, here's the idea here with pre-symptomatic diagnosis. Some genetic disease genes that don't really show up until later in life have certain DNA fingerprint patterns that are associated with them. Okay? These are late onset genetic diseases. There are some genetic diseases that babies are born with. There are some genetic diseases that don't show up until later in life. Much later. And if a person has that particular gene, mutated gene that would lead to that genetic disease, in some cases there are DNA tests that can be done, and that person can find out ahead of time whether or not they have that gene. Now, whether they want to know that or not, that's another bioethical question. But I want to look at a couple examples here. Does anybody know who that is?
Woody Guthrie.
Good, Woody Guthrie. Woody Guthrie. Born in 1912, died in 1967 at the age of, 55. Are we ready to start? All right, we're ready to start. Happy Wednesday. Again, thank you for coming back. Because if you didn't come back, I wouldn't have a job. So, appreciate it very much.
Such a well-paid job.
Yeah, right, but it's fun. It's good to see you guys. And what I want to do here is I'd like to do a little bit of, I'd like to channel Mr. Rogers a little bit. Okay, it is a beautiful day in the neighborhood. But I want us to do a little, let's go to the land of make-believe. Okay? Let's make believe for a minute. Let's make believe that I'm a scientist. I work in the FBI DNA fingerprinting lab. And we're working on a case, a very sad case, where a young woman was murdered, long blonde hair. There were a few long blonde hairs at the crime scene site. And they have a suspect in custody. It's a guy with kind of medium length red hair. All the crime scene investigators found at the scene of the crime was one single red hair. That's it. They were very careful. I mean, they swept the scene. One little hair. And now, okay, it's a red hair. Now, the suspect in custody, he's got red hair, but that's no big deal. Lots of people have red hair, right, Mary Helen? Okay. All right, sorry about that. Maybe I shouldn't have gone there. I mean, a lot of people have red hair, so what? The suspect has red hair. We found one single red hair at the crime scene. So what? I mean, that's really not enough evidence, is it? Well, there weren't many cells at the base of that red hair. Just a few cells. There's not enough DNA in those few cells at the end of that red hair. There's not enough there to put them on a gel like we talked about last time and run a DNA fingerprint. There's just not enough DNA there. Now, there is a particular target sequence of DNA that we could look at as a length of DNA that all people have. It's non-coding DNA. It's not part of a gene that makes a protein. It's non-coding DNA, and there's lots of variation in the sequence of A's and T's and C's and G's in that stretch of DNA. So if we had enough of that DNA, my goodness, if we had enough, we could run a DNA fingerprint, and then we've already run a DNA fingerprint on the guy that's in custody. Then we could compare the two and see if they match up. There's just not enough there. So basically what we've got here is, okay, this is a little plastic kind of a test tube with a cap. That tube's only about that long, real small. And in that tube, there's some liquid in there. It looks like water, but it's actually, we call it a buffer solution. It's got some ions in there. It's not just pure water. And we've put the crime scene sample in here. It's only just a handful of cells. It's not enough to really do much, but there's something we think we might be able to do with that. Okay? What we'd like to do, if we could, in the lab, is make billions of copies of that little sequence of DNA. Boy, if we could do that, if we could make billions of copies of that, then we'd have enough. We could put it on an electrophoresis gel. Remember electrophoresis? And run a gel and look at a DNA fingerprint if we only had enough. Well, now, there's a way to do that. It's called PCR, which is the title of today's lesson. There's a way to do that. What's PCR stand for? Polymerase. Polymerase, that's the enzyme that builds the new strands of DNA, that replicates DNA. Chain reaction. Because if you get that thing started, you're going to start with, theoretically, one DNA molecule that replicates, and now you've got two. And then they each replicate, and then you're going to have 4. And they each replicate, you're going to have 8. And 16 and 32 and 64. They're going to reproduce in number exponentially, explosively over time, because they double every time. And so you're going to end up with something called a chain reaction once it gets going. These primers, they act like bookends. They bracket, they mark specifically. That's the only part right there that we're going to replicate. And you've got a picture in your handout of that process, step by step. Looks like this. So this is what it looks like. This is what's going on in that little test tube in the lab. Okay, up here, let's set the stage. Here's our DNA molecule, double-stranded. See, it's like a ladder, twisted ladder that's been untwisted for the picture. And in the lab, to do PCR, this is what we're going to do. Now, oh, by the way, this is the target sequence. That little piece, out of all the DNA, that little piece that we want to make billions of copies of, that's the target sequence. So, we've put all the DNA from those cells in that little tube, all of it. Okay? But the only part that we're going to make copies of is this little target sequence. So the primers, there's a primer there, they attach and they bookend that target sequence. We pick out specific primers that will specifically bookend or target that sequence. So here's the process. You heat that sample up to 94 degrees Celsius. Boiling is 100, so that's almost boiling. And what that does, look at that, it denatures, separates the 2 strands. So here's our 2 strands of DNA separated. Then we've got to cool it down to 60 degrees Celsius. And when it's cooled down to 60, that allows this primer to attach here, this primer to attach there, and it also allows the Taq polymerase molecules to latch onto the primers so they can get ready to replicate. Okay? But Taq polymerase works best at 72 degrees Celsius, so you got to heat it back up to 72, and then when you do that, this Taq polymerase molecule, click, 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 goes along, replicates that DNA. This one over here is replicating that one. Now we've got 2 Identical DNA molecules where we only had one before. See that? Got one only. Got two where we only had one before. That's one cycle. All right, we've got to repeat that in the lab 39 more times. Yeah, so what we're doing with PCR is we're just making billions of copies of a target sequence. Now what we do then when we run the electrophoresis gel, and she showed you that kind of what I talked about last time, you just squirt all the DNA in there, including all the genomic DNA that belong to that person or whoever, like if you're trying to do a DNA fingerprint on somebody's DNA. But the only DNA that's going to show up in a band on the gel is the target sequence, because we've made billions of copies of it, so there's enough there that it's going to show up on the gel. So remember restriction enzymes last time? Restriction enzymes aren't involved in this stuff at all. Okay? We're not cutting up DNA, we're just targeting a little certain segment that we're interested in, and that's what we're making copies of and then running in the gel. Would you like to see some applications of how this has been used? There's all kinds of applications. So I just want to share just a few, okay? Ways that it has been used. Pre-implantation genetic diagnosis. Okay, if you were in any of my sessions last fall, we kind of touched on this, so let's look at what that is. Okay, a little bit of review. That's a uterus. Remember the uterus? Half a uterus in the picture. Ovary, fallopian tube. Okay, so you've got an egg released from the ovary. If a herd of sperm cells shows up and one gets into the egg, you have a fertilized egg, it goes through its first mitosis division, becomes a 2-celled embryo, and then 4, and then 8, and then 16, and then 32, 64, pretty soon a little almost microscopic ball of cells, hollow ball of cells that implants in the lining of the uterus. Almost a week later, then you've got a pregnancy. Okay, that's how it happens in vivo, right? But some people who have maybe the female partner has blocked fallopian tubes or scarred, and the egg can't get through, and the sperm can't swim up through here. They might have to do in vitro fertilization, where mom's eggs are put in the dish, petri dish in the lab, dad's sperm cells are added to the dish, fertilized outside the body, in vitro. And then if you get a good embryo from that, then they can put that embryo, it's almost microscopic still, back up into the uterus where hopefully it will attach. That's in vitro fertilization. There's a technique where PCR is employed in this whole process in cases where they need to check to see if the embryo has inherited a bad disease gene from parents. Okay, here's an example of where that's been used. Now, what we're looking at through the microscope here is a photograph of an 8-celled human embryo in a petri dish in the in vitro clinic. 8-cell human embryo. Why are they testing it? Because there's a very bad genetic disease called Tay-Sachs disease, where it's caused by a single called a recessive gene. And if mom on her 2 chromosome number 15s has a good normal gene on 1 chromosome and a bad Tay-Sachs gene, mutated Tay-Sachs gene on the other one, she's a carrier, but she's normal because she's got the good gene, which is dominant, kind of cancels out the effects of the bad gene. Now she's either going to give her baby this chromosome or this one. If she gives her baby the chromosome that has the bad gene on it, and if her husband is also a carrier, and he gives the baby the chromosome that has the bad gene on it, then that baby's going to have a double dose of the bad gene that causes Tay-Sachs disease. And if that baby survives pregnancy, embryo development, that baby's going to die 100% of the time by the age of 3 or 4 from a horrible downhill digression. It's heartbreaking. It's awful. It's awful. It's called Tay-Sachs disease. Okay, so here we're looking at a case where the couple that went through IVF, they're both carriers. They're both normal, okay? But they're carriers. Mom carries the gene, dad carries the gene. And they want to test this embryo to see if it has inherited 2 copies of the bad gene. So what they'll do here is, with a tiny little microscopic glass needle, the tip is microscopic almost, they'll suck out, look at this, with that needle, apply suction to it, see it? Sucking out 1 cell, plucking 1 cell off of that 8-celled embryo. Now that doesn't hurt that little 8-celled embryo because it's so early in its development, it doesn't matter. All 7 of the cells that are left are identical. And the DNA that they have is identical to the DNA in that cell that they've removed. So removing that 1 cell, there it is, that doesn't hurt that 7-celled embryo. They're going to let that 7-celled embryo sit in the petri dish long enough, and it'll be okay in there for a while, to where they can take that cell that they've removed, that single cell, do PCR on it, on the specific Tay-Sachs gene. Make billions of copies of that gene, run it on a gel. If it's the bad gene, it's going to show a certain fingerprint pattern on the gel. If it's good gene, it's going to show a different kind of signature on the gel, fingerprint pattern. You with me? This is how can be done. Now, that's only a single cell, not much DNA in there. Not much DNA in there, but with PCR you can make billions of copies of the target sequence. If it turns out that the DNA in that cell, if that cell has 2 copies of the bad gene, what do you think they're going to do with the 7-celled embryo? Let it die. Not implant in the uterus, because all the cells are identical. If it turns out that this cell is either a carrier or totally normal, then they'll take that embryo and put it back, put it in the uterus, hope it implants. That's another application. How many of you have ever taken a little paper strip test to see if you've got COVID? Have any of you ever? Yeah, okay, lots of people. Now that strip test works pretty well. That at-home test works pretty well, but there's one that's a lot more expensive, that your sample has to be sent off to a lab and they can do something called PCR testing on it. Okay, it's a more sophisticated, more sensitive test. Nod your head yes if you've ever heard of the PCR test.
What is PCR?
That's what I was just getting ready to ask. I was just getting ready to say nod your head yes if you've ever heard of PCR. Do you remember PCR last time, vaguely? Sort of. It's a way of taking a small DNA sample in the lab and making lots and lots of copies of it so that you have enough DNA material to work with, like if you want to run it through an electrophoresis gel and do a DNA fingerprint. Okay, that's PCR from last time. The take-home message, all we have to remember about PCR is it's a way of taking a really small sample of DNA in the lab and making really billions of copies of it. So we have enough that we can do something with it. Okay. So we had to do PCR last time so we can look at how the PCR test works today. But let's look at the virus. And that's what it's called, severe acute respiratory syndrome coronavirus type 2. SARS-CoV-2. And it's actually a picture of it's kind of pretty. Now that's a computer animated, AI generated image of a COVID virus. It's got all these little, they're called spike proteins on the surface. We'll look at why those are important in a little bit. All those spike proteins on the surface of the virus give it kind of a halo or crown or corona appearance. That's why it's called coronavirus. And these are the guts. And you've got, I put a lot of pictures in your handout so you don't hardly have to worry about taking a whole lot of notes. Just kind of follow along. But this virus has several parts. Now, you see that green part, that dark green, round dark green part on the outside? That's a lipid membrane. Now, the outsides of your body cells are lipid membranes. So it's a lot like cell material or cell membrane material. It's a lot like that. It's a lipid membrane. So it's basically a hollow sphere of fat molecules, lipid molecules. But then it's got all these different protein molecules embedded in it, and they all have different jobs as far as maintaining the integrity of the shape of the virus, allowing the virus to invade your cells and get inside. And so several different protein molecules. One of the most important ones is this spike protein right there. These brown lollipops sticking on the surface, they're called spike proteins. We're going to find out what they do here in a little bit. Spike proteins. They're really important. Well, they're all important. The other thing I want to point out is inside the virus, there's a single strand of RNA. And that RNA molecule is divided up into several different genes. And the thing only has 11 protein coding genes. That's it. Instructions for how to be a virus, how to act like a virus, how to invade cells and take over and cause infection. That's it. Now, it's kind of interesting how we humans, we have 20,000 pairs of coding genes. This thing only has 11. I'm sure you've heard of the HIV, the virus that causes AIDS. It only has 9 genes. So there's not much to them really in terms of their size and their complexity. But those are the parts. So here's the key thing. It doesn't have DNA, it's got RNA instead. Single-stranded RNA. And that's kind of how the virus is put together. We're going to talk about some of those parts and why they're important here as we go. All right, so what's the virus do? This is one of your cells. That's one of your cells right there. It's the only part of it. The cell's really big compared to the virus. The virus approaches, and these spike proteins, what they do is they hook up to little receptor proteins that you already have on the surfaces of your cells. Those little receptor proteins are kind of like doorknobs on the surfaces of your cells, and the virus latches on and kind of opens up that doorknob and then gains entry into the cell. It doesn't knock, it just goes right in. Okay? Uninvited, obviously, it enters. Now, What's it do after it enters? Okay, here's your cell, one of your cells. When the virus enters, it releases its RNA. That's its single-stranded RNA right there. And what kind of instructions are in that virus RNA? Instructions for how to replicate itself and how to brainwash your cells. It kind of brainwashes, hijacks the cell, takes over the cellular machinery, and basically hijacks the cell and at gunpoint says to your cells, all right, you're going to stop what you normally do now and you're going to start manufacturing millions more COVID viruses. And when these are all manufactured using your own cellular machinery, all these components of these viruses, then they're going to be put together, and we're going to make millions of new viruses inside this cell, and then they're going to leave the cell, millions of them, and go and infect other nearby cells, and the infection is going to spread in the body, okay? This picture is top of page 2 in your handout, okay? And then I've listed the detailed steps. Now this is a lot to swallow at one time. So I thought if I give you this handout, then you can take it home, and in your spare time for fun, you can study the handout and the steps that are listed there right below this picture. So let's just kind of talk about this a bit. Okay, this is one of your cells. There's a cell membrane. Here's a COVID virus attaching. The specific receptor is called ACE2, but we don't have to remember that. And this enzyme, TMPRSS, that enzyme helps this receptor. The virus gets in. Step 1, the virus enters. Step 2, it releases its RNA. This thing that looks like a slinky. That thing that looks like a slinky, that's a single-stranded RNA molecule. It's got 11 coding genes in it. It's released. Step 3, some of the RNA instructions are used to make proteins that will make up a whole bunch of new viruses that are going to be manufactured by this cell. Some of the RNA is going to stick around and it's just going to be replicated to make more RNA molecules for more viruses that are going to be manufactured. Step 5, all these components, proteins and RNA that's going to make up new viruses, they're all packaged and put together in the Golgi apparatus. Step 5, see, that your cell is manufacturing more viruses, and then they emerge. Step 6, they emerge, they leave the cell. Millions of them leave a single cell and go and invade other nearby cells, and the infection spreads throughout your body. The types of cells mainly affected this way: respiratory tract cells and intestinal cells. Sometimes people, when they get COVID, they have diarrhea also. All right, that's how that virus operates. And the reason the virus RNA, those virus genes, mutate so often, so regularly, and we keep getting new variants, is because RNA, when it replicates, it's not as efficient as when DNA replicates. In our DNA, when it replicates, we've got DNA proofreading enzymes that check to make sure if there are any mistakes, they're corrected. Our DNA is very efficient when it replicates. DNA is very efficient when it replicates. And so, when RNA replicates, when a virus's RNA replicates, it doesn't have proofreading enzymes. or RNA repair enzymes. And it makes mistakes more often and they're not caught. So when an RNA gene replicates, it's sort of like if you're typing real fast on a word processor and you never do a spell check. Okay? What comes out is full of mistakes. And that's why we keep getting new variants. Here's how the PCR test works. And I've given you a lot of diagrams and step by step. We have to kind of remember PCR a little bit. So here's a nasal swab. Oh, I always hated that. The nasal swab, but there it is. And if this has mucus on it or saliva, let's say, from an infected person, there could be 100 billion little viruses on that swab. Here's one enlarged COVID virus. Basically, remember, just a collection of proteins with virus RNA inside of it. Okay, we're going to take that swab, that sample, back to the lab, and there's the RNA inside of it. And what we're going to do in the lab, what are we going to do with that sample? We're going to run PCR, but before we do that, in order to do PCR, we've got to have DNA, right? Yes. And this is virus RNA that we've extracted. We can't do the PCR on the virus RNA. So fortunately, in the lab, we can take this virus RNA, if it's present in the sample, and we can make virus DNA, which is just a DNA copy of that virus RNA. And we do that by a process called reverse transcription. Now, we have to flash back to a previous session. Let's do that. Remember this? Y'all remember this? DNA, this is what your DNA does, makes RNA. That's called transcription. Then the RNA is read by a ribosome to build proteins. That's called translation. That's the central dogma because that's what all living things do. But in the lab, we can make it go backwards. Watch. We can take RNA and make it go backwards and make DNA. That's called reverse transcription. We can take that RNA sample, if the person was infected and there's RNA in the sample, we can take that RNA sample in the lab and we can make DNA, a DNA version of the virus's instruction manual. It's called reverse transcription. And to do that, we're adding an enzyme called reverse transcriptase. That's the name of the enzyme that allows us to take the virus RNA in the lab, make it go backwards and make DNA. And then that DNA is called cDNA or copy DNA. Now, what kind of instructions are in that double-stranded DNA? The same instructions that were in the virus's RNA. 11 coding genes, only it's in the DNA form. Now that it's in the DNA form, we can run PCR on it and make billions of copies of that virus DNA. Now, what's going to happen if the person was not infected and there's no virus RNA in the sample, and we add the enzyme reverse transcriptase, which is going to make RNA into DNA? What's going to happen if there was no RNA in the sample to begin with? We're not going to get any cDNA, right? We're not going to get any. And if we run PCR on that to make billions of copies of that cDNA, we're not going to get any product. There's not going to be any cDNA there. How are we going to know? I'll show you how we know. You remember PCR, you heat the sample up, the DNA strands unzip, they denature. There they are. And then you cool it down and you add the primers and the polymerase enzymes that replicate the DNA. Is this a little bit familiar from last time? Okay? We're doing PCR on that sample, but this time we're not going to, and now we've got 2 copies of the virus DNA if there was virus RNA in the sample. We've got 2 copies of it. We're going to do that cycle maybe 40 times, make billions of copies of that cDNA, that virus DNA, but we're not going to put it into an electrophoresis gel. Instead, what they do is they attach these little fluorescent molecules to the DNA. Little fluorescent glow-in-the-dark molecules. And while this is running, real time, that's why you may have heard it called real-time PCR. While the PCR is running, they have an optical detection instrument set up to measure the fluorescence or the glowing of these little fluorescent tag molecules. As more and more DNA is being produced, it glows brighter and brighter in the optical detection instrument, and that's how they detect it. And if they see these glowing tag molecules accumulating in real time as the PCR runs, then they say, oh, we got a positive test here. So here's what happens. Let's jump down to the bottom of this picture here. Let's pretend that the sample came from somebody who's not infected, but they wanted to get tested. Okay? If they're not infected, and they do reverse transcription to take any possible virus RNA in the sample and make cDNA, virus DNA. If they're not infected, if there's no virus RNA, there's not going to be any cDNA. And if they add the primers and the fluorescent tag molecules and the polymerase enzymes to try to run PCR on there, if there's no cDNA, if there's no virus DNA, there's not going to be any billions of copies of virus DNA made with fluorescent tags, and so no fluorescence is going to show up, and that's going to classify as a negative test.
Well,
If the virus, if the COVID virus is mutating constantly, doesn't that mean that the primers would also have to be adjusted for the various varieties of COVID?
Yeah. If the virus is constantly mutating, they have to keep updating, developing new primers, and that's why it's so important to keep on funding this stuff and keep it going. Let's look at how an mRNA vaccine works. It's kind of cool how it works. Here's what's in an mRNA vaccine. It's actually pretty simple. An mRNA vaccine is pretty much just a little hollow sphere of fat molecules, lipid bilayer, a double layer of lipid molecules, a little hollow sphere of these lipid or fat molecules with a little piece of messenger RNA inside of it. Not all 11 coding genes of a virus are in here. Only a little small piece of RNA that has instructions in it for how to make the spike protein. Remember the little lollipop knobs on the outside of the virus? That's the only protein that is coded for by this short little RNA molecule. So that's really all an mRNA vaccine is. Now I say all, I mean it took decades and decades to develop mRNA vaccines. Took a long time, lots of work, lots of research. Everybody in science stands on the shoulders of giants, right? And so that's what it is. And they're processed and they're bottled and so on. So the spike proteins, again, these little knobs here. So out of all these proteins, and there are others that I didn't even label, out of all these proteins, the only one that's coded for by that little tiny piece of messenger RNA inside the vaccine is the one that says build spike proteins. Okay, so when a person receives the mRNA vaccine, these little vaccines, molecules, they're calling that. Little nanospheres of fat molecules with a little piece of RNA inside, messenger RNA. Those little fat spheres fuse with your muscle cells. It's injected into the muscle. They fuse to your muscle cells, and also immune cells that are in the area of the injection. You got immune cells all over in your body, but they'll fuse to the outside of those muscle cells and immune system cells, and they release that little spike protein messenger RNA into the cell. It does not go into the nucleus. Messenger RNA only knows how to go out of the nucleus. It doesn't know how to go into the nucleus. So it goes into the cytoplasm, all this jelly-like material in the cell, and it attaches to a ribosome. All ribosomes know how to do is read messenger RNA molecules and follow the instructions and build whatever protein those instructions say. That's what our ribosomes are doing, right? All the time, building protein molecules. And if the instructions in that little piece of spike protein RNA say, make spike proteins, that's what that ribosome is going to do. So here's a string of amino acids that protein's made of. The ribosome strings together these amino acids. It folds up into this three-dimensional shape. There it is. It's a spike protein. What happens to that messenger RNA after the ribosome reads it? Same thing that happens to all your messenger RNAs after ribosomes read them. It degrades. The cell breaks it down, degrades it. The spike proteins come out here to the edge of the cell. They're released out of the cell, and antibodies and other immune cells recognize those spike proteins, and it's like they freak out. They say, these are foreign proteins. We need to recruit the troops. We need to get immune system cells to start reproducing to make more. We need to start making specific antibodies that can seek out and destroy these foreign proteins, these spike proteins. Now, that really primes the immune system, you see. That's what this vaccine does. Primes the immune system. Creates kind of a memory in the immune system. The immune system gets all fired up and all excited, starts producing all these special cells that know how to make these antibodies that know how to attach to spike proteins, and so then they're all in place. They're all in place. So that later on, if this person gets invaded by the actual virus, those actual viruses and invader are going to get ambushed because the immune systems have been primed and the numbers increase, the reinforcements increase, and they're all ready. And so when the virus invades, the real virus, later on, it's either going to be totally wiped out and the person's not going to get sick, or the person might get COVID, but it'll be much, much less severe. Remember when General Custer and his soldiers were ambushed and wiped out by the Native Americans at the Battle of Little Bighorn? No, we don't remember that. We've read about it, right?
Yeah,
We read about it. Do you suppose there were some Indians or Native Americans that died in that battle? Yeah. And there were soldiers, American soldiers who died, but the numbers of Native Americans vastly outnumbered the soldiers. That's kind of how this works. If you had the vaccine, your immune system cells and antibodies far outnumber, and they're ready to ambush any viruses that happen to invade later. And they don't need to recognize the whole virus. Your immune system doesn't need to recognize the whole virus. All it has to be able to recognize are these spike proteins. Developing the mRNA platform, or the basic structure of an mRNA vaccine, is like developing a wanted poster. And once you've got that poster developed, you can slip any bad person's, any villain's picture in there, right? And you can come up with new vaccines for new diseases as they come along very quickly. That's the advantage. I mean, the basic structure of mRNA vaccines, the basic platform, took decades to develop, and it's ongoing as they try to improve the platform. But once you've got it, then you see a different face can be put into that wanted poster, slotted in easily. And so when novel viruses come up in the future, and they will, then we can quickly make new vaccines by slipping in whatever mRNA molecule we need to slip into that vaccine in order to make it effective. Now, I was playing around with this little analogy that they have here of the wanted poster. I have to go back to the computer to show you this. I was playing around with it, and as I was playing around with it, after I read this, I said to myself, I think I get it. I think I get it. Let me just show you what I mean here. Okay. You have no idea how long it took me to develop this wanted poster platform. You have no idea how long it took me to develop this mRNA vaccine, okay? I mean, the poster, with the artwork and the font and all, took a long, long time. And so I decided, okay, I'm going to stick an mRNA molecule in there for some disease virus, okay? So here it is. Here's the mRNA molecule. I stuck this one in there. Okay, so we've got a wanted poster. And hey, I think it took maybe it's because I'm too much of a perfectionist. It took me about an hour and a half to design the wanted poster. How long did it take for me to just stick my picture in there? No time at all. In fact, look what we can do. Now that we've got the basic platform, the basic structure of an mRNA virus, look what we can do. We can stick almost anybody in there. Okay? We can put anybody in there. We could take Beyonce and put her in there. See? Or, I mean, this takes no time at all. But to build a poster, or we can put Taylor Swift, or any number of Anderson Cooper. See how it works? You could put a Neanderthal person in there. Haven't seen one of those in 2,000 years or so. We could put Joe Biden in there, or we could put Donald Trump in there. There's put a lot of people in there. So it's real easy. Once you have the mRNA ready to go, the platform, the mRNA vaccine, it's easy. If a new novel virus shows up, sequence its RNA, slip it right in the vaccine, and you're good to go. It started October 1st, 1990. It was predicted to be completed by September 30th, 2005. It was going to be a 15-year project. Take a long time to do that. And what's interesting is it actually finished ahead of schedule and under budget. Now, the United States carried the majority of the workload, and it was a federally funded project. Now get this, okay? Let this sink in. A federally funded project that finished ahead of schedule and under budget. That never happens, but it happened in this case. And the reason it happened this way, ahead of schedule, the reason it happened was as time went on, computer automation improved dramatically between 1990 and 2003. As the project went on, the computer automation got better and better, and faster, and that's why it finished ahead of schedule and under budget. So that's why. These are the main countries that worked on this project. Lots of them. Now they're in alphabetical order. I put them in alphabetical order there, so don't worry, the United States is right there. Okay, USA. Carried the bulk of the workload. Now, the director in the United States, the director of this project was this man, Dr. Francis Collins, and quite a resume. And that project spanned the Clinton and George W. Bush administrations. So they were ultimately his boss. Okay? He was also then director of the National Institutes of Health from 2009 to 2021, director of NIH. Now, why did they pick Collins in the first place? He had a vast resume. He had discovered the cystic fibrosis gene previously. He had discovered the Huntington's disease gene. Quite a resume. And so that's why he was picked to head up the project. So, director of NIH under 3 presidents. Worked under 3 presidents. 1, 2, 3. Can you see the pictures? Okay, 3 presidents. Obama, Trump, and Biden. And then finally science advisor to the president for part of 2022 under Joe Biden. That's kind of his background. But he was the director of the Human Genome Project. Now, Francis and I go way back. Actually,
Photoshop.
no, it's not Photoshop. It's real. That's back in 2007. Francis Collins came to Purdue, and in Stewart Center, he gave a lecture, a free lecture, open to the public. Well, it was open to Purdue people and open to the public. And so I thought, I'm going to go to that lecture. And I told my genetics students, if any of you go and take notes and bring those back, extra credit. So I had a bunch of kids go to this. And in fact, the kids, a lot of them bought his book that came out just right before that. And I bought it too, entitled The Language of God. One of the public kind of, for the general public books that he wrote. And it's a great read. If you haven't read it, I highly recommend it. And so some of my students showed up with their copies of the book, and they got to meet him, and I chatted with him briefly, and he autographed my copy of his book, and it was a moment. It was a moment. A rock star in the world of genetics. I mean, literally. I went to a convention in Atlanta, the National Association of Biology Teachers, and he was like one of the main speakers, okay? Big auditorium, okay, got all these biology teachers from all over the world in there. And at the end of his lecture, he pulled out his guitar, and he sang and played on his guitar, and he sang a song about the human genome that he had written. And all these biology teachers start putting their phones up with the lights on and crowding up to the stage, and he literally is a rock star. So, for sure. Okay, but anyway, this was the goal of the Human Genome Project. They used the tools of molecular genetics, restriction enzymes, sequencing, some of the things we've talked about, to decipher the complete DNA recipe for humans, and they also did other organisms at the same time. All kinds of other living things, not just humans. All right, so what did we learn from this? I just want to go over some highlights. We learned that we have 20,000 gene pairs, not 100,000. The previous predictions, in fact, I learned this in college, was we probably have 100,000 gene pairs because we're made up of at least 100,000 different kinds of protein molecules, right? A gene makes RNA, makes a protein. So the prediction was we probably have 100,000 gene pairs. The Genome Project revealed that, no, sorry humans, you're not as not what you think you are. You only have 20,000 gene pairs, about the same number as mice. Mice have around 20,000 gene pairs. Well, now obviously the instructions in them are different than the instructions in our genes, but some of the instructions are the same. Some of them are. Corn, depends on what variety of corn we're talking about, 32,000 gene pairs. Now, if you want to talk chromosomes, there's a little shrimp that has like 254 chromosomes. And you and I have 46. So, anyway, an interesting discovery. Now, how does that work? How do you make, at least 100,000 different kinds of proteins, and you only have 20,000 gene pairs? How do you do that? One of the things that was discovered was this process here called alternative splicing. Okay, let's set the stage here. All right, this is a gene right here, from here to there. A sequence of DNA, and it's divided into different parts. These 1, 2, 3, 4, 5, those sections of that gene are called exons because they're sections of that DNA that are going to be expressed. The instructions used to build protein later. And then in between those exons are these intervening sequences called introns. And what happens is the DNA right there, it makes RNA there. But before this RNA leaves the nucleus of the cell and goes out and attaches to a ribosome, remember how a ribosome will read that messenger RNA and build a protein? Before it leaves the nucleus of the cell, that RNA right there is processed. It's processed. The introns are cut out and thrown away. The exons are glued together. Now, here you're looking at how one mRNA molecule from one DNA molecule has managed to produce two different mature processed mRNAs. Okay? In one case, exon 1, 2, 3, and 5 are glued together to make a mature mRNA molecule there that will be translated to produce one kind of protein. Does this make sense? And sometimes another kind of mRNA can be made from this, where 1, 2, 4, and 5 are glued together to make a second kind of messenger RNA. So from one gene here, in this example, we've got 1, 2 different kinds of RNAs that can be made from one gene. So we've got instructions in that gene to make really 2 different kinds of proteins. So it's not just one gene, one protein. In this case, it's one gene that can make 2 different kinds of proteins, depending on what the cell needs. So that's one thing we learned. Another thing we learned is most of our DNA does not code for proteins. Most of it. Only 2%. What? Only 2% of our gene codes for proteins. So what's the other 98% doing? Oh my, this is kind of humbling in a way. You've got this pie chart in your handout. Okay, look at this. 42% plus 8%, 50% of our DNA is actually virus DNA. Virus DNA. It's DNA from viruses that invaded our ancestors over millions of years, inserted their own DNA into our genomes. They don't do us any harm. After millions of years, they have, quote, figured out how to not harm us. And all they do, all that's left are these little bits of DNA embedded in our DNA. And every time our cells replicate the DNA, they replicate those little bits of virus DNA all along. That's all the viruses, for millions of years, have, quote, wanted to do, is reproduce. And so they use us to replicate their DNA. Oh, here's another interesting thing that was discovered as a result of the Human Genome Project. The DNA similarities between species reveal an unbroken, without gaps fossil record. It was striking what was discovered. Because remember, not only did they sequence human DNA, but DNA of a whole bunch of other organisms or living things as well. Now let's look at some examples of DNA similarities. Okay, if you compare one human to another, their DNA is 99.9% the same. Now, that means 0.1% accounts for our uniqueness as individuals. 0.1%. Now that amounts to 3 million bases. So 3 million letters accounts for our uniqueness. Any 2 humans on the planet, you compare their DNA. If I take DNA from a long distance runner, Olympic runner from Nigeria, isolate his DNA in a test tube, analyze it, sequence it, and I take Mary Helen's DNA, your DNA compared to his, 99.9% identical. And humans and chimps, 98.8%. Our DNA compared to chimpanzee DNA, 98.8% the same. A, T, C, G, and so on. Humans and dogs, 94%. Humans and cats, 90%. Humans and cows, 80%. Humans and fruit flies, 60%. Humans and bananas, 60%. All right, here's another related to the evolutionary story, kind of another surprise. We still carry broken genes. They're called pseudogenes. You and I have a bunch of broken genes that we still hang on to and pass on generation after generation. They're called pseudogenes. They're genes that don't work anymore. They're broken. But they're not broken in other closely related species. But they're broken in us. We have about 200 to 300 pseudogenes. That's the essence. Broken genes. So, the question that I've thought about is, were we purposely created with broken genes that don't work? Or could we have inherited them from ancient ancestors? But anyway, let's look at this. Here's one example. This is like my favorite example of a pseudogene that we have. It's called the gallunolactone oxidase gene. It makes a protein called gallunolactone oxidase, which is an enzyme. An enzyme that controls one step in a chemical reaction in our bodies that leads to the production of vitamin C. And we have that gene. But it's broken in humans. It's broken in us. It doesn't work. So we don't make that enzyme. So that means we can't make within our bodies vitamin C. We have to get it from food that we eat. Like oranges, grapefruits, lemons. I don't normally eat lemons. Limes, citrus fruits. Okay? Why do we have to eat citrus? Why should we eat citrus fruits, or why should we at least take vitamin C vitamins? Because we can't produce it in our bodies, because that particular gene in us is broken. We inherited it from our parents, we passed it on to our kids, and so on. It's in all humans. It's a broken gene. It doesn't work. So anyway, in us, okay, there's the gene. In us, it's broken, so it's called a pseudogene. The result, no vitamin C. So we have to consume vitamin C. Now, what happens if you don't have enough vitamin C in your diet? Ah, that's right, you guys are with it. Scurvy. Here are some mammals, other mammals, that have that same gene, but it's not broken in them. It's not broken in these guys and these guys and these guys. Pigs, cows, cats, dogs, mice. Other mammals, they have that gene and it's working. It's producing vitamin C in their body, and so that's why, like, I've never seen my dog eat an orange. I've never seen a cow. They might, but they don't. They don't eat citrus. They don't need citrus fruits. Okay? What are some examples of mammals that have that gene in them that's broken? Well, chimpanzees, gorillas, orangutans, and humans. So in other words, that gene broke long before we split off into primates in this. And you know what? When I did this, I wasn't surprised by the ancestry that was revealed, the countries that my ancestors came from, because we kind of have that written down in our family over generations. But what I was surprised to find out is that I too, for example, I have like 240 Neanderthal variants. in my DNA. And so I've wondered, maybe this could be my 2,000 times plus great-grandparents. Homo sapiens, Neanderthals, maybe. I'm just making that up. But it's something like that. If our ancestors did interbreed with Neanderthals. Oh, we're going to finish up with this one. All humans, this is another discovery, 99% identical. We mentioned that a little bit ago. Only 0.1% accounts for individual differences. Only 0.1% makes him look different than her. 0.1%. All humans. The racists of the world, when they found out that all humans on the planet, no matter where they come from, are 99.9% identical, freaked out and said, no, that's fake news. That can't be true. We are all one family. In fact, so much so that there's actually more genetic variation within a race than there is between races. And so biologically, strictly biologically, there's no such thing as race. We're all on a continuum, not discrete, quote, races. Race is a social construct. All right, and finally, and this is on the last page of your handout, and I'll let you read that on your own. The most important surprise that came out of the Human Genome Project was all these discoveries that have enabled modern medicine to accelerate in all new developments related to cancer research, personalized medicine. The future is exciting. It's ongoing. And there's work. The Human Genome Project didn't just stop. The work is ongoing now. It's part of genomic research that's still going on. Revolutionizing medicine. And we need to stop right there. That's all. That's all there is. The final has been canceled. All right, now, let's do this. It's not totally random, I know. It's like fake random. Let's do a random generator program here to figure out who's going to get this random drawing of this major award. Okay. All right, check out the number in your upper right-hand corner, written in red ink on your handout. The number. I made up 75 handouts. Raise your hand if you do not have a handout. Oh, there's somebody over here who does not have a handout. Do we have any more? Here's one who doesn't have a handout. Wayne doesn't have a handout. Oh my goodness, we don't want to leave anybody out.
Do you else need a number?
We don't want to leave everybody have one? All right, so let's see. All right, I'm going to put in here that we're going to run numbers between 1 and 75. It's too small for you to see. You're going to have to take my word for it. Are you ready? I'm going to click the button and generate a random number. Kind of random. Okay. 25. You have 25? We have a winner. We have a winner. And what are you going to get? You're going to get your own copy of this book. You're going to get your own copy of this book. It's a major award. It's not a leg lamp. It's better than a leg lamp. Okay. Anyway, I just wanted to give away one of these because, tell you what, it's not a New York Times bestseller. Anyway, congrats. All right. So